Our next speaker is Associate Dean Hu Zhibin. Dr. Hu is an MD PhD. He's Associate Dean of the School of Public Health at Nanjing Medical University, where he is also a professor in the Department of Epidemiology and Biostatistics. He received his training as MD and PhD from Nanjing. He was a visiting scientist for the Department of Molecular Epidemiology at the German Cancer Research Center. He is vice chair for several prestigious associations in China, including the Chinese Medical Association, the Professional Committee of Epidemiology, and the Chinese Preventive Medical Association. Let's give him a warm welcome.
Thank you, Professor Post. I think it is not it's a little bit too late and not a right time to talk about basic science, right? My first thing to present is my great pleasure to come back because I visited Professor Zhang just 2 months before, yeah. So it's very great pleasure to present in UCLA School of Public Health. My talk is about our recent genetic analysis of hepatitis B virus susceptibility and also that is HBV and also HBV-related liver cancer. My lab is the epidemiological branch and genetic co-facilities for the State Key Lab of Reproductive Medicine and also Cancer Center. Okay, as many people may know, HBV persistent infection is the prevalence is consistent with the prevalence of liver cancer. So HBV infection actually is the most important risk factor for hepatocellular carcinoma, except Japan. Japan is high for HCV, but also high for liver cancer. And the risk factors for persistent HBV infection include age-at infection, male gender, alcohol use, and also co-infection with other hepatitis viruses. But the most important thing is age-at infection. Adults can be infected with HBV, but will recover very quickly, completely. So, also other studies suggest that host factors also play important roles in HBV infection. So there are some genetic analysis. In 2009, a Japanese group conducted first genome-wide association study for persistent HBV infection. Their discovery phase included about 800 cases and then 2,000 general controls. I highlight this because these people they didn't know the HBV infection status. But you know, if they get infected, some of them could be persistent infected. So these controls actually have some misclassifications. So I think this kind of design could a little bit reduce the power of screening. But they still find some letter SNPs around the HLA molecules. That is HLA-DPA1 and HLA-DPB1. These two letter SNPs are very close. To extend their findings and also to see if there are some particular loci among Chinese, we conducted the genome-wide association last year. The cases are HBV persistent carriers, and the controls are subjects who had naturally cleared HBV after acute infection. And of course, all of them are HCV negative, that is hepatitis C virus. But hepatitis C virus are very lower among Chinese. This is the design. Here we can see cases of HBV persistent carriers. We can see here. And the controls, there are HBsAg negative, HBsAb positive, HBcA positive. This means they're really infected. And HCVAb negative. And all these people are vaccine-free. The GWAS scan include about 1,000 cases and 1,000 controls. Then we have 22 suggestive SNPs. After two internal validations, internal means people from the cases and the controls from Jiangsu province. We have 3 survival, and then 2 external validations. We still have these 3 ones. This is the first letter SNP around the HLA-DQB2. Actually, one of the letter SNP is exactly the one Japanese people identified. This means, although the study by Japanese have some potentially misclassifications, they still can identify a very strong signal. And also, there are 3 ones around this region. But by our studies, we find 2 more. One is close to HLA-C. This is type 1 HLA molecular. This means not only type 2, because HLA-DQDP is type 2 molecular. HLA type 1 also play important roles in HBV persistent infection. Another one is a non-HLA. This is located in the last intron of UB2L3, actually identified by other studies, immunity-related studies. This is the ubiquitin-related molecular, and the latest step is the EQTL loci of this gene. The A allele means the risk allele is associated with the higher expression of this gene. And this could result in maybe the accelerate the degradation of the NF-kappa-B molecular, because this is ubiquitin related. Then attenuate the immune response to HBV infection. Sorry. Okay. When we change the case control situation, we use HBV-related HCC patients as cases, and also HBV persistent infections as controls. We also can evaluate the genetic susceptibility of liver cancer, hepatocellular carcinoma, after persistent HBV infection. With similar design, we have 2 significant SNPs related to the liver cancer susceptibility. The first one, interesting, also between HLA-DQA1 and HLA-DRB1. This means type 2 HLA molecular plays important role not only in HBV persistent infection, but also people get persistent infected. They're important in the determining of the outcome, the cancer. Another one is located within the first intron of GRIK. This is involved in the glutamate signaling, but we didn't do further evaluations for this pathway. We also persistent, another genome-wide association study also have independent screening in China, actually did by Fudan. They also identified 2. The first one, same with us, HLA-DQ, and then another one is around STAT4. Because STAT4 is important in immune-related pathways, So their paper get published in Nature Genetics. After these findings, I think one very important thing is discover missing heritability, because both HBV persistent infection and also liver cancer, we still have very few loci. So we can do some international collaboration by pulling GWAS data via consulting and sharing pipelines and samples. Now we are just putting the two independent screening from China together, and hope we can find some new. Then another thing is genome-wide based sequencing for rare variants and also copy number variants, but this still costs too much. So one thing we can do is to find out synthetic association. This means if we really think HLA is important, just use HLA region based on haplotype analysis or EQTL analysis. You do the file mapping, sequence-based file mapping to find synthetic association. This would be cost less than genome-wide association, genome-wide sequence. Here is an example for file mapping. We have GWAS tagging snippets around here. Then we have candidate gene region. This could be haplotype analysis or EQT analysis. Then we can do in silico annotation based on public available database like ENCODE, TCGA, to find EQTL and also methylation QTL loci. Then we capture all the regions, both coding regions and regulatory regions, then sequence them. Okay. But for sequence-based targeted region, after file mapping, you still have a lot of work need to do. For example, you have a lot of peaks for the letter SNPs, around the letter SNPs, a lot of peaks. And they are independent. Based on conditional logistic regression analysis, haplotype analysis, likelihood ratio test, you need to do large sample size to separate variants displaying the greatest associations. And this beta use different ethnicity samples. Then functional analysis of candidate elements is required. Now popular with genome editing by like Cas9. Also, after the functional analysis of candidate elements, you need to functional analysis of candidate variants, like a report gene, chip sequence, iron binding assay, and also like chromatin conformation capture, 3C. Then also other cultured cell lines and human tissue studies and animal models. Zebrafish and Drosophila is not very good for cancer studies, so you need a mouse and maybe other animal models. So for me, from an epidemiological view, I think at least equal important, that is gene-environment interaction. A new wave of GWAS based on carefully designed large representative nested case control study using stable internal exposure instead of external exposure may be needed, because these studies already done, dramatically of them are pushed by geneticists, are not epidemiologists. So you are hard to pull them together because the environmental information is not so comparative. Here I give an example because virus host interaction, liver cancer is a very good model. So you can see here we doing the HBV, the virus subgenotype typing. The below piles are all controls we can see from different regions from Jiangsu province. You can see the subgenotypes of HBV is just varied from region to region. But if we also genotyping the cases, we can see here, the C2 genotype are higher than 60%. So C2 genotype is consistently higher among cases. Here we can see all below 40%. So HBV subgenotype actually a very important risk factor, together or independent from host genetics. When we get the host variant in, we just use one here. This one is identified by our GWAS, actually the HLA region. You can see when people have the C subgenotype and also the risk allele, they have more than 10-fold increased risk and very clear interaction. Now we are doing multi-center validations on this data. And also, next step, we are think about the whole genome-wide genetic and host and virus genome interactions. And not only genotype, the subgenotype of the virus, but also sequence them. Okay, acknowledgement. Thank you for a lot of institutes, and I only listed the PIs because the page limit. They are great contributions and efforts for this project. Thank you very much.
We're running a little late, but we have time for one question. Yes, please.
Hi, my name is Zhuang. I'm a faculty of school medicine. I'm also an alumni from our school of public health back in 1990. That's why I'm here today.
Thank you.
And also I'm from China too, so I saw this excellent composing put it together. I'm really excited. And public health is a big issue in China, and even though I left China many years ago, I still pay attention to what happened in China. Thank you very much all to the faculty and the deans here today. My question for you is, my mother is a hepatitis B carrier. There was the reason she had to catch that was when she was sick, was hospitalized in China about 30 years ago. She had a transfusion of serum. That time is very popular. When people get sick, their body get weak. In the Chinese, in China, and then one of the treatments to give some serum to give some nutrition or some protein to improve their immune system, immune function. I know that time there's no screening, and all the serum is pulled from many donors, and that I know all these donors have no screening for their hepatitis or any viral infection or any infectious agent. Now my question is, why after so many years the incident of hepatitis is still that high? I know China still is getting better now in terms of screening all the donors. Thank you.
Okay. Very good question. Actually, the horizontal transmission of the HBV, I think, is get fewer and fewer in China now because the cell blood cell is forbidden, and most of the infected ones I mentioned, age of infection is important. Most of the infected ones actually get the virus from their mother, and now we're using vaccine and also like active immune when they're born, the situation is also better. But here I present all the infection situation for the adults. So the infection situation is still very high. But you know, for young people, for like 10 or below 20, the infection rate is getting very low. And also liver cancer. Now the liver cancer not decrease, still high, because for HPV infection and the liver cancer, there are a long time to develop. But for the young people, the incidence of liver cancer also getting down. Yeah.
Thank you.